Hello everyone. Today I would like to talk about the paper The antiviral PDGR49 provides protection from lethal neurotropic Venezuelan equine encephalitis virus intranasal infection in mice. For the key concepts, I would like to go over the direct and indirect infection. For direct infections or intranasal route of exposure, virus would infect olfactory bulb, particularly olfactory epithelium and olfactory neurons. For indirect infections or subcutaneous route of exposure, virus would infect osteoblast, Langerhans cells, dendritic cells, and fibroblasts. From there, they would travel to the secondary lymphatic system, such as lymph nodes or spleen, and cause infection peripherally. The virus invades a central nervous system by enter through the circumventricular organs of the brain with lacked a blood-brain barrier. However, the blood-brain barrier is still intact upon central nervous system entry and does not lose entirety until peak viral replication in the brain. After virus get into the brain, they would infect central nervous system target cells like neurons, endocytes, microglia, oligodendrocytes. Infection of these cells activate microglia and endocytes, which stimulate the transcriptions of interferon-stimulated genes and pro-inflammatory cytokines. And during peak viral replication within the brain, the blood-brain barrier becomes compromised. The combinations of viral replication and inflammation would cause neurodegeneration, leading to neurological complications. Therefore, treatment of encephalitis viruses require fast-acting drugs. And currently, there's a limited set of antivirals for neurotropic infection and none for alphaviruses. To address this gap, researchers tested the antiviral PDGR49 using a mouse model in vivo preclinical study. They found out that the viral load of V-infected mice treated with PDGR49 suggested improvement. So by looking at the two graphs, we see that the infected virus and viral RNA were reduced 1 to 2 logs in the brain of V-infected PDGR49 treated compared to V-infected group without treatment. In histopathology experiment, they compare the images of the mock group, the sham group, and the infected group received PDGR49 treatment and harvested olfactory bulb at day 4, day 8, and day 14, and they found out that in the mock group, there's an intact histology structure, no V glycoprotein. In the V-infected group, there's abundant lesions, V glycoprotein, and numerous activated microglia. In the group that received PDGR49 treatment, there's a reduced amount of V glycoprotein, minimal neuronal destruction, and increased numbers of activated microglia at both day 4, day 8, and day 14 post-infection. At day 14 post-infection, no neuronal degeneration, minimal V-lipoprotein, and lower activated microglia compared to day 8 post-infection. And here is another image of the V-lipoprotein shown in red, and they compare between the two groups, the infected group and the infected group received PTGR49 treatment. Obviously, there are very few V-lipoprotein at day 4 post-infection in the brains in the PTGR49 treated group. Main conclusions of the research: PTGR49 showed a promise for the treatment of mice exposed to VIV. Practical implications: PTGR49 addressed the gap in treatment of viral encephalitis. For future research directions, there should be an improvement of broad-spectrum antiviral efficacy of PTGR49 through structure, activity, relationship studies. I hope that you enjoy the talk, and thank you for your attention.